Hello, and welcome to the second lab presentation. Today, we're going to be covering the material that's listed and identified in Lab Exam 2 Study Guide. And let's quickly just go through and see where we're going in this presentation, what areas we're going to cover, what concepts we need to know. So at the very beginning in this study guide, you can see that we're going to cover something, this concept of homeostasis. We'll talk about active and passive transport and osmosis. The core concepts that that this whole lab is designed around is, again, homeostasis and this idea of form and function, which we talked about, and osmosis. Then I've given you a series of statements. So pay attention to these. These are the kinds of statements that I would develop questions around. So you should be familiar and know this material by the time this lab is ended or after you have studied this for a while. There are some more additional questions here, as you can see. There's a reading that you should be doing, page 65 to 74 in your lecture textbook. All right. Also, please note that I've also given you a list of terms that you should know. So starting with osmosis, isotonic, hypotonic, hypertonic, excuse me, solution, passive and active transports, physiological saline. Talk about that when we get into the experiment. And some additional terms. You'll see a lot of repeat in these lists. That's okay. Just be aware that they're repeats, and I'm just emphasizing these terms again. So in this case, though, you need to give me examples of the same terms. So here would be definitions and then specific examples of these what these terms represent. We'll also be looking at some passive transport processes, so how things are moving across the membrane, and active transport processes. We're not going to spend a lot of time here. We'll just talk about these very briefly, and then we're going to spend most of our time in this osmosis, and we'll talk a little bit also about simple diffusion, facilitated diffusion, and carrier-mediated facilitated diffusion. And then there are some more terms and concepts that you need to be familiar with here. And finally, on the next page, some additional terms. So these are terms that describe, the first three are terms that describe the shapes of cells. And this is where we get into function, form and function. And as I said, we'll cover this in our experiment. And then we'll also need to understand what this means, physiological saline. All right, so that's where we are. Let's quickly go through active transport first, get that out of the way. Then we'll talk a little bit about some passive transport processes, and then we'll be zero in on osmosis. All right, first of all, what is the difference between a passive transport process and an active transport process? And it's very simple, really. So with passive transport processes, all of these, no energy is used by the cell for these things to happen, for things to move. With these processes, there is no energy expended by the cell, so no ATP is being used. In these processes, this requires energy. On the part of the cell. In other words, ATP is going to be used. So all of these things you should list here, that you see listed there, require the expenditure of energy by a cell. Let's take a very, very brief and quick look at what these are, and then we'll forget about them. We'll talk more about them in lecture. So first of all, let's talk about the primary and active transport. And I'm not going to spend too much time on that, but let's go ahead and take a look. So here's an example of active transport. And as you can see, we're going to move something from one side of the cell to the other, and we're going to use ATP to do that. So we're going to use a molecule of ATP converted to ADP. We're going to break off that last phosphate group. So we go from adenosine triphosphate to adenosine diphosphate. And in that process, we're releasing energy. That energy is used to move this from this side of the membrane to the other side of the membrane through that protein. Okay? So generally speaking, when we talk about active transport processes, what we're really talking about is moving something against its concentration gradient. So on this side of this graph, what you're looking at is a series of different molecules: green, gray, yellow, purple, and they are all concentrated on this side of the membrane. And they're not very common or very concentrated on this side of the membrane. So that creates a concentration gradient from high concentration to low concentration. And if you're moving something using active processes, you're moving something from a low concentration to high concentration. You're moving against the concentration gradient, and that's why energy is being used in that process. So active transport requires the use of energy. Now, as you noticed perhaps in that list, there are a couple of other things. So I'm not going to talk about the difference between primary and active transport, but in both cases we're using ATP and moving something against its concentration gradient. But in this case here, we're doing something slightly different. It still requires energy. So endocytosis, exocytosis, transcytosis, and phagocytosis. Let's take a quick, very brief look at what that's all about. And here's an example of a process of endocytosis. So if you, let me zoom out so we can see a little bit more of this. So this represents the membrane of a cell. And what we want to do is there's some things that are on the outside of that cell, and we want them on the inside of that cell. And the way we're going to do that, is we're going to have them, in this case, bound to a protein on the cell membrane. And then we're going to form a sac. We're going to invaginate the membrane. We're going to pinch that off, and we're going to bring that into then to the cell and do whatever it is that we want to do with that. So this is a process called endocytosis. It's bringing something into the cell. Endocytosis. We can do the same thing in the opposite direction. Now, this is not an example of that, but let's, for the sake of argument, say that the process is we built these little yellow balls inside the cell, and we want to move them to the outside. We're going to form them, or we're going to collect them in a vesicle, and that vesicle then will merge with the membrane, and then we will just simply have those on the outside of the cell. So they will exit the outside of the cell. So whether you come in, endocytosis, or go out, exocytosis, requires energy. There are a couple of other ways that this can play out. For example, phagocytosis is basically the same thing, except we're talking about a very large structure now rather than smaller things. So something like a microbe or something like that. So we're going to bring that in. Again, we're going to use a membrane sac, capture that, bring it into the cell. In this case, we're going to digest it. So phagocytosis is to digest something once it's brought into the cell. But all of this requires energy. So all these processes, whether it's primary or secondary, endocytosis, exocytosis, transcytosis, or phagocytosis, all those require energy. Now, just very briefly, if you're wondering what transcytosis is, what's going to happen is we're going to take something and bring it in at one end of the cell, transport it across the cell, and release it on the other side. So in one side, out the other. Trans, going across the cell. Okay. So endo, in the cell. Exo, out the cell. Trans, across the cell. And phagocytosis, phago means to eat, and what they're referring to is a large thing like a bacteria. All of these require energy. So let's talk about these these concepts here, the passive way in which we can bring something into the cell. And in those situations, in all of those situations, what we're looking at is moving something down its concentration gradient. So here we have a high concentration of these molecules. Here we have a low concentration, and we're going to move these things from high concentration to low concentration. And the first one on our list was simple diffusion, which is being illustrated right here. So it's just going to pass right through that membrane. But let's define the term diffusion. So let's do that. Let's define that term. And then we can move on and talk about the other processes. So diffusion, what is the sort of scientific definition of this term diffusion? Diffusion is a passive process in which there is a net or greater movement of molecules from a region of high concentration to a region of low concentration until equilibrium is reached. This is something that you're quite familiar with, and it happens all the time around you. You take advantage of this all the time. If you make tea, if you put on perfume or aftershave lotion, you're depending on diffusion to either fill the hot water with the tea, or to allow that perfume or aftershave lotion to whiff off of your body and capture somebody's attention. All right, so let's very quickly draw a little example of what that would look like. So if we have a container of some sort, doesn't matter what kind it is, and we have or we get a concentration of some molecule. Right. So that concentration, now we can see that it is highly concentrated in this corner and not so much concentrated in this corner. And then what's going to happen is we're going to get movement from this high concentration to the low concentration. So we have a concentration gradient. So we have high concentration to low concentration. Very simple concept to understand. Now this can happen in a gas such as the atmosphere. That's why you can smell a skunk after you've passed it on the road. It can happen in water, such as when you put a tea bag in hot water and it gets filled up. So let me show you an example of that. Here is a liquid example of diffusion. So we have a blue dye of some sort concentrated here at the bottom of this test tube. And then as time goes on, you can see that that blue dye is moving from high concentration to low concentration until equilibrium is reached. That is a random and evenly distributed dye throughout the body of water. Okay, so that's an example of diffusion. All right. So diffusion. Let's take a look. Let's go back and look at that a little overhead that I had and come back and look at the idea of simple diffusion. So here we are, simple diffusion. We have high concentration, low concentration. We're moving from high concentration to low concentration. In this case, we're moving across the membrane. So that membrane is defining two spaces, the outside of the cell and the inside of the cell. And that has high concentration, this has low concentration. So it's moving down its concentration gradient. All right, well, that's simple diffusion. What about some of these other processes? Facilitated diffusion, for example. What is involved there? Or carrier-mediated facilitated diffusion. Here we're talking about the same concept, except we're going to use a protein to move something across the membrane. So we can use a protein, and this would be channel-mediated facilitated diffusion. Or we can use a carrier protein. So this would be carrier-mediated facilitated diffusion. So these are all examples of facilitated diffusion. In this case, facilitated diffusion using a channel or a tunnel, if you will. So the protein does not change shape. It's simply acting as a door through the membrane to allow something to move across it. And in this case, which is carrier-mediated facilitated diffusion, this protein has to change shape. So whatever it is binding to that protein. When it binds, the protein changes shape and moves that molecule across the membrane. All of these things that you see here do not require energy. Energy is not being required, as we see here, with active transport processes. Okay. All right, so now that we have a grasp of some of these basic ideas, transport processes, simple diffusion, let's focus in on a more specific type of movement, passive movement, which is osmosis. And our lab is about osmosis, so let's define it. Osmosis. Okay. Osmosis. All right, the definition. The net movement of water molecules through a selectively permeable membrane from an area of high water concentration to a area or region of lower water concentration until equilibrium is reached. So the net movement of water molecules through a selectively permeable membrane from an area of high water concentration to an area of lower water concentration until equilibrium is reached. Now, if you think about that definition for a moment, it sounds very, very similar to our definition of diffusion. We're moving from high concentration to low concentration. So that's the same. But what are the differences? Why isn't this diffusion, simple diffusion, I should say? And the answer is that there are two critical and important differences in the definition of osmosis and the definition of diffusion. Let's for a moment come back to the definition of diffusion. So here it is, a passive process in which there is a net or greater movement of molecules from a region of high concentration to a region of low concentration. So here, there's no definition of what those molecules are. They can be anything. Anything you want, as long as there is a difference in concentration. And there is no mention of a selectively permeable membrane. So if we come back to this definition, what we see is that now what's moving is something very specific. Water is moving, and it's moving through a selectively permeable membrane. Okay, so something very specific is moving, and it's moving through a structure, a selectively permeable membrane. What is a selectively permeable membrane? Well, they come in a variety of different shapes and forms, but they all have one thing in common. They're permeable, but only selectively. So some things can pass through them, and other things can't. That's the selectivity part. They are permeable to some things, and they're not permeable to other things. In our class, in biology, what we're talking about when we talk about a selectively permeable membrane is the cell membrane, the plasma membrane of a cell. That is selectively permeable. It is a membrane through which some things can pass through and other things cannot. Okay, so selectively permeable. Now, another way that we can, excuse me, let me go back and let's list the differences. So, two important differences. We're talking about water is the specific molecule moving. That's one difference. And two, it's through a selectively permeable membrane. Two critical and important differences between this osmosis and diffusion. So what's going on? Why is this? Why are we talking about water? Why not some other molecule? And the way to understand that is through a very simple demonstration of the of what we're dealing with. So we're going to be talking about solutions because we're dealing in biology and in the human body specifically. And so let's take a look at this example. So I'm going to draw, this represents a selectively permeable membrane. Okay. And it separates two solutions. So we have a solution over here, and we have a solution over here. Okay, now we've used the term solution. What is a solution? Let's take a look and define what a solution is. A solution is composed of two things, a solvent plus solutes. That's the definition of a solution. And we covered this in lecture. A solvent, in our case, is going to be water. And something else dissolved in it. Salts, let's say it, salts. Sodium chloride. Okay, those are my solutes. Okay. So, if we come back to this example now, I said we have a solution on this side and a solution on this side. And we also know at this time that we have a solvent plus solutes here. And we have a solvent plus solutes here. And we're further going to stipulate, I should say, that the solutes, which we're going to do in blue, okay, are more concentrated on this side of that selectively permeable membrane than on this side. So we have a concentration gradient, right? More blue things over here and less over there. Now, under the rules of simple diffusion, what you would expect to happen is that these solutes, the blue things, would move from high concentration to low concentration. But they can't, because this selectively permeable membrane is preventing them from moving. So simple diffusion is not going to allow, or there's not going to happen, because the selectively permeable membrane will prevent that from happening. So that leaves the solvent. The solvent will have to move. Water is going to have to move. But now we have a problem. How do we figure out which side of this selectively permeable membrane has more water? All we know at this point is that there's more solids over here than over here. If I poured a liter of fluid in here and added that salt, and poured a liter in here and added a little less salt, I would still have, according to what I just told you, a liter of water here and a liter of water there. So how can I have a difference in concentration? I'm going to answer that, but there is an easy way to think about this, which I'm going to show you right now. So the easy way to think about this is to remember that this is an equation, a solution, that this is an equation here. And I have a body of, or a solution on this side and a solution on this side. And so the easiest way to figure out if there's a difference in concentration of water is to use this equation. So I'm going to call this side A, and I'm going to call this side B. Okay? And now I'm going to plug in this equation for both of those solutions, A and B. So solution equals solvent plus solutes. Okay, that's for A. And for B, I can write out the same equation. The solution equals the solvent plus the solutes. Okay. Now we're going to put some numbers in this. So I'm going to come back to my example here, and I'm going to give you some numbers. I'm going to say that this is a 30% solution. That is a 30% saline solution. And over here is only a 5% saline solution. Right? So the saline solution is referring to the solute. So I have 30% here in terms of the overall solution and 5% over here. Okay, so now that we have these numbers, we can come back to our equation and start plugging in some numbers. First of all, if this plus this equals that, then this always has to be 100%. So we can fill in this side of the equation, because by the rules of mathematics, if two things equal 1, then this is the whole. That's 100%, right? So, and now do we have another number? Yes. So for A, I have 30%. That's the solutes. So 30%. For B, I have 5% solutes. So 5% solutes here. Now, all I have to do is solve for the unknown. I'd take 30, subtract it from 100, and I get 70%. I take 5, subtract it from there, I get 95%. And I must ask now, I ask myself, do I have a difference in concentration in solvent? And indeed I do. You can see that I have, at this point, I have a difference in concentration. So I have 95% here and only 70% there. So that means that this is greater than that. So what is the rule in osmosis? If you go back and look at the definition of osmosis, we're going to move water. The net movement of water molecules through a selectively permeable membrane from an area of high water concentration to a region of lower water concentration until equilibrium is reached. Okay? So from high concentration to low concentration, water is going to move in that direction. From B to A. From B to A. So coming back, then we can predict, based on what we know about osmosis, that water is going to move in this direction. Okay. That's how I want you to analyze problems that I might give you in any test. Is I will always give you the solutions and concentrations and percentages so that you can easily do the math. But it doesn't really answer the question because, as we mentioned before, I put 1 liter here and I put 1 liter here. Why does that change? Why does that change the water concentration when I add the salts? More salts over here and less over there. And without going into too much detail, let me go ahead and show you a little picture to help you try to understand how that is different. So over here, again, this is my A and this is my B. So I'm reproducing what we had up above. And now I'm going to kind of magnify something and really make it big so we can see it more clearly. And I'm going to take these things here and I'm going to say these are my solutes. Okay. All right. Those are my solutes. And what's going to happen is this is sodium. I'm not going to put in the chloride, but you'll know it's there. As you can see, I have more chloride over here, more sodium over here than over here. So that still is in keeping with what we had said earlier. And now what's going to happen is that these water molecules are going to associate with these sodium. So if I have 10, well, let's make it 10. If I have 10 water molecules over here and 10 water molecules over here, and each one of these is going to have 2 water molecules associated with it, okay? So 2 and here, because remember that water molecules are going to associate. They're going to form those polar bonds. So I have 1, 2, 3, 4, 5, 6, 7, 8 water molecules bound up to sodium here and only 2 here. So that leaves me 2 water molecules over here and 8 water molecules over here. These that are bound to the sodium are out of play because they're attached or connected to the sodium, and so they're not free to move. But the water that's not bound up to the sodium can move, and the water that's not bound up to the sodium can move. And as you can see, there is less water bound up over here and more water not bound up over here. So I have a greater concentration of water on this side than I do on that side. So, but that's expressed in these percentages that you see here. Okay, so this is why this happens. This is how you analyze and find the difference in concentrations. Okay. All right. So let's apply this now. Let's apply this idea to some more concrete examples and bring in some terminology as we do that. So let me collect myself here and get rid of the stuff before I'm overwhelmed with everything. All right. So at this point, you should have one of these in front of you. So you should have downloaded this. This is the Osmosis handout. And before you came here, you should have downloaded this. If you haven't, then go ahead and put me on pause. Go out and find this, download it, and bring it back, and then continue. I'll give you a moment to do that. Okay. So that moment is now up. Let's begin. So we're going to apply what we've just learned now to 3 different conditions. So what I've drawn here is a square with an oval inside, and obviously that is some compartment with a solution in it, and then a cell is sitting inside that compartment. And I'm going to stipulate that I have a 10% solution here and a 10% solution here. So the concentration of solutes in the water surrounding the cell is the same as the concentration of the water in the cell. So there's no difference in concentration. And by now, you should probably figure out what's going to happen here. So remember, inside the cell, I have 100%. Outside the cell, I have 100%. 100, not for 10%. Okay. And I have 10% here. And I have 10% here. And if I do my math correctly, that's 90%. 90%. Is there a difference in concentration? No, there's no difference in concentration. And so what we can say now is there's going to be no net movement of water. So no net movement of water. Why? Because we have a selectively permeable membrane separating 2 spaces, and the concentration of water in both those spaces are equal. So I have the same amount of water here as I have here, because the percent solution is the same. So there's no net movement of water. So what we can say is that the cell is isotonic to the solution. The cell is isotonic to the solution. What does isotonic mean? Tonic, we're going to say, refers to molecules. Now, technically that is not true. Tonic doesn't really mean that, but for us, that's what we're going to say. Iso means the same. So same molecules, same number of molecules. The cell has the same number of molecules as the solution. We can say the same thing for the solution. The solution is isotonic to the cell. We have the same concentration of solutes here as we have on the inside. We have the same concentration of fluid on the outside as we do on the inside. Okay? All right, so what we have then, this is a situation in which we have an isotonic solution. So whenever you hear that term, I have in my beaker an isotonic solution to whatever is in that beaker, typically a cell of some kind. What I am telling you is that the concentrations in the solution and in the cell are the same. And I'm comparing always the solution to the inside of the cell. So isotonic solution, this is isotonic to that. Okay. Now we get to describing the shape of the cell. And what we're going to say is that the cell is normal. The shape of the cell is normal. Okay. If we're talking about a red blood cell, which we will be in our experiment, this is a red blood cell. And as you can see, the red blood cell has a particular shape. So that shape is called biconcave. So in the case of a blood cell, we can say this is biconcave. Only in the case of a red blood cell. Other cells have other shapes. But if we put red blood cells in an isotonic solution, what we'll have is a cell that has a normal shape or its normal biconcave shape. So this is what we're talking about. Notice this little indentation here and over here, little indentation there. What is that all about? Well, what happens is these red blood cells are transporting oxygen to your body tissues, from the lungs to your body tissues, and back from your body tissues to your lung is transporting carbon dioxide. So most of the oxygen that you carry in your blood is being transported through red blood cells. And these red blood cells are passing through the lung very, very quickly. Extremely fast. And we're talking about a second or two that it stays in the lung, and then it's out of the lung and around and through the rest of the circulatory system. So in order for that cell to pick up oxygen, to become saturated with oxygen, it has to be very efficient. And one of the ways it can do that is increasing the surface area. So by indenting the surfaces here, I create more surface area. More surface area allows me to have more diffusion, and it's more efficient for that molecule to enter the cell or leave the cell if the cell is in the area of your tissues. Okay? The other thing that shape gives me is flexibility. So when this cell passes through the little tiny capillaries, it has the ability to be flexible because those capillaries are extremely small. And sometimes these red blood cells have to pass through a capillary single file. They're so small. So the shape gives me increased surface area and flexibility. So the shape then is connected to its function. So remember, form follows function. So in the case of red blood cells, that biconcave shape is important to the way it functions. And if you lose the shape of a red blood cell, if it's no longer biconcave, then what happens to function? It doesn't function. It doesn't function the way it's supposed to. And so you're going to lose you're not going to be able to transport oxygen, for example. So we're going to tie this all together now. Form follows function. So remember that at the beginning we talked about the concepts, and find that very quickly here. There we go. The core concepts of this lab. Homeostasis and form follows function. We've just demonstrated that in order to keep the function of this cell, we have to place it in an isotonic solution. If that cell is in its normal shape, it has its normal function, and we can maintain homeostasis. So the form of cells is directly related to homeostasis because in their proper form, they can do their job and help you to maintain homeostasis. And that's how these concepts are connected. And we're talking about that within the context of osmosis. Okay? So form follows function, homeostasis. And the connection there is that by maintaining a normal shape, we maintain the function of that cell, and we thereby maintain homeostasis of the body. Okay? All right. Well, let's go on to another example. So set this aside and see if we have another example here. And indeed we do. So I'm going to now stimulate a different set of circumstances. I'm going to say I have 15% here and 10% here. Okay. By the way, let me very quickly, I might as well do it at this point in time. I want to talk about a concept that I mentioned earlier, which is physiological saline. So I'm going to talk about physiological saline. We talked about normal shape. Okay. Now I'm going to talk about physiological saline. So when we have this condition, isotonic solution, have equal concentration on the inside as and the outside of the cell. So what I can say is 10% is my is the physiological condition inside the cell in terms of the concentration of solutes. So this then is a physiological saline. Saline refers to salts. Physiological saline solution means that it's isotonic with the cell. So if I tell you a 10% solution is a physiological saline solution to your body cells, you immediately know what the concentration is inside your cells. It's the same, 10%. Okay? Oakley dokleys. Let's come back and talk about this next example. So we have 15% solution on the outside of the cell and a 10% solution on the inside of the cell. Now we want to figure out if water is moving, and if it is, where is it going? So we come back to our equation. We say 100% there. We already know that. 100% there. So this is inside the cell, outside the cell. All right. And let's fill in the numbers. So inside the cell, we have 10%. Outside the cell, we have 15%. Now we just solve for the equation. And this gives me 90%, right? This gives me 85%. Okay. Is there a difference in concentration? And as you can see, there is a difference in concentration. I have a greater concentration there on the inside of the cell than on the outside of the cell. So where is water going to move? It's going to move from inside to outside. It's going to move from inside to outside. Okay. Everybody see that? All right. Now, let's introduce some terminology here. I want to describe the cell compared to the solution. And what I see when I look at the percentages is that's 10%. That's 15%. This is less than that. So the term that I'm going to use is hypo, meaning less or under. Hypotonic to the solution. The cell is hypotonic to the solution. Okay. The solution, 15% compared to 10%. That's greater than that. So what is the term? Hypertonic. The solution is hypertonic to the cell. Okay. Hypo meaning less, hyper meaning more. We know that water is moving from inside the cell to the outside of the cell. Okay. So it's moving from inside the cell to outside the cell. Water moves from a hypotonic solution to a hypertonic solution. So we can add that up here. This is hypertonic. This is hypotonic. Water is moving from hypotonic to hypertonic. Okay. Now the question becomes, what happens to the cell shape? Water is leaving the cell. And just like water leaves a grape when you leave it in the sun, it shrinks, it shrivels. It becomes a raisin. But we don't want to use shrink or shrivel because that doesn't sound very scientific. We want to have a nice scientific sounding term, and so the term we're going to use is this: crenated. It means that the cell has shrunk. But shrunk doesn't sound very scientific. It doesn't make us sound very smart. So if we want to sound smart, we say crenated. What happens to the shape? It's changed. What happens to the function? It's changed. What happens to homeostasis? Not going to happen. The cell cannot contribute to homeostasis because its shape has changed, its function has changed, can no longer function properly, and can no longer contribute to homeostasis. So what we can say is the shape is crenated. So the function is lost. Non-functional. Cannot maintain homeostasis. So we can describe now the solution. The solution is hypertonic. So in a hypertonic solution, cells will crenate. Okay. Let's give one more example. So we'll come back, get one more of these, and we will now set up a different example. So I'm going to say I have now a 5% solution here and a 10% solution here. We're going to go ahead and solve our equation for the inside and the outside of the cell. Okay. So fill in our numbers. The inside of the cell is 10%. The outside of the cell is 5%. That means I have 90% here. And I have 95% here. Is there a difference in the concentration of the solvent, of water? Yes, there is. As you can see, this has a higher concentration. So water is going to move from this to there. Water is going to move from the outside of the cell to the inside of the cell. So outside, in. Okay. So how do we describe that cell? Well, if we look at the cell, it has more than the outside. So the cell is compared to the solution is hypertonic. The solution is less, concentration is less outside the cell compared to the inside of the cell. So that is hypotonic. And water is moving from the outside to the inside. Okay. So water is moving from the outside to the inside. And what do we say? The solution is hypotonic. This is hypertonic. And once again, we see that water is moving from hypotonic to hypertonic. Let's talk about cell shape. What happens to the cell shape? Well, most of us, hopefully, have, when our youth, have played with water balloons. And so what happens to a water balloon when you put too much water in? It breaks. It bursts, right? But once again, breaking or bursting doesn't sound very scientific, so we want to try to find a term that is scientific. And we're going to use this term, hemolysis. Hemo refers to red blood cells. Lysis is to break apart. Okay? So if you're talking specifically about red blood cells, then you can use the term hemolysis. If you're talking about any other cell, it's lysis for red blood cells. Okay? Well, obviously, if the cell has broken apart, then function, non-functional, non-functional cells cannot contribute to homeostasis. They cannot contribute to homeostasis, and they will not even exist. So, what kind of solution is this? The solution is a hypotonic solution. Hypotonic because it's less than the inside of the cell. Okay. All right, let's take a look at some examples of this. And here is, this is from your textbook. So isotonic solution. In an isotonic solution, there's no net movement of water. The cell maintains its shape. So we have a normal red blood cell shape. And because it has a normal shape, it has a normal function, and it can contribute to homeostasis. In a hypotonic solution, that's the one we just talked about, water is going to move into the cell, it's going to break apart, it's going to be non-functional. In a hypertonic solution, what you're going to see is a shriveling or a shrinking of the cell. It's going to crenate. So it's either going to hemolysis or crenate. The only time that you get a good result is if you have an isotonic solution. That is a physiological saline solution to the red blood cells. That's the only time you're going to get good results. Anything other than that, and you have a problem. The cell function disappears, is disrupted, and you don't, or you can't maintain homeostasis. All right. Well, that gives us the background necessary now to understand our little experiment. So let's walk ourselves through that experiment and then see what happens. Now, normally at this point, if we were in a face-to-face class, we would have test tubes and some solutions in front of us, and we would be able to add the material to the solution. I just happen to have some examples of that. So we would have a setup that looked like this. So we would have 3 test tubes, and we'd put solutions in them, and then we'd add blood, and then we would observe them under the microscope. Whoopsie daisy. So there we would make a slide that looks like this. Okay. Now, my expectation had been when I began thinking about this is that I would be able to show you the results of this experiment using my microscope, but I just discovered over the weekend that somehow my microscope, the substage condenser, is out of alignment, so I cannot use it to demonstrate this, which is unfortunate. I have to bring in Olympus rep to figure out how to fix that, because I can't seem to figure out how that got out of alignment or how to get it back into an alignment. All right, so we're just going to walk ourselves through this. So we have 3 test tubes, and we're going to put in different solutions. We're going to put in distilled water in one solution. So distilled water is literally 100% water, no solids. And in the second test tube, we're going to put in physiological saline. So immediately you know, because it's physiological saline, that that is a specific type of solution. Hopefully it's popped in your head. I'm not going to say it now because this is part of the questions in the next set. So we're going to get physiological saline in test tube number 2. And then in test tube number 3, we're going to put a high solute concentration, 9%. Okay. All right. So, by the way, notice that in this second test tube, we said physiological saline, and the concentration is 0.9% sodium chloride. That's physiological saline solution. All right, so then we're going to do some observations. And if you had done this experiment, you would have noticed a couple of things. So with the distilled water, that is the first test tube, what you would see is a clear solution. There would be no opaqueness to it. It would clear up. It would be red. There would be a red color, but it would be very, very clear. And the next 2 test tubes would be dark or opaque with some slight difference between them, which I'll explain in when we go through the experiment later and talk about the results. Okay, so in this test tube number 3, it would be slightly more viscous, so it would be thicker if we looked at it. Okay. All right, then we'd observe it under the microscope. So what is your expectation at this point? If I observed the solution here, if I observe the solution here, and if I observe the solution here under the microscope, what is my expectation? Let's first ask ourselves, let's predict which one of these test tubes, when you look through the microscope at the cells, which one would you see have normal shaped cells? Which one would contain cells that look like this? Let's go back and analyze what we've added to each of the test tubes. So in test tube number 1, distilled water. Test tube number 1, physiological saline. In test tube number 3, we had a high concentration. So what do you suppose would happen? Let's make our predictions. In test tube 1, test tube 2, test tube 3. Okay. So, what I'd like you to do right now is put the presentation on hold, take a moment and think about this, and then come back and we'll see if your predictions meet or are the same as my conditions. So I'll go ahead and let you take a break now, and I'll just go tiddly-dee, tiddly-dum. Here I am waiting for you. Come on back. Let's start all over again. And we're on. Okay, test tube number 1. What's your prediction? We put distilled water in it. Okay. Well, if we wanted to think about this, we could subject this to the same analysis that we had done earlier, right? So we could come back and use our handy-dandy little handouts and do the analysis with this. Hopefully some of you thought about doing this. So, distilled water, and so inside here, what percentage do we have? 0%. We have pure water. No solutes. What do we have inside the cell? Well, I didn't tell you directly, but I think you can figure it out. Remember, test tube number 2, we added physiological saline, and then I told you that was a 0.9% solution. So if you do the logic, if you carry the logic, and physiological saline is 9%, so if that's physiological, if that's the number for physiological saline, that means inside the cell we have 0.9%. 0.9%. Okay. We go through our analysis. So inside the cell, outside the cell. So inside have 0.9%. We have 0% here. That makes that 100%. And here I have 99.1%. If I do my math right, I'm not a mathematician, that looks right. Okay. So do I have a difference in concentration? I do indeed. Perhaps not very much, but it's still a difference. There it is. So water's going to move in this direction, is it not? It's going to go from the outside of the cell to the inside of the cell. Water's moving in this direction. So the cell is hypertonic. The solution is hypotonic. Okay. And the cell shape, ladies and gentlemen, is homolysis, because we are talking about red blood cells, so we can use that term. What about function? Non-functional. So this is a very bad situation if you happen to have this happen to you for some reason. Very unfortunate if that's the case. I'll say goodbye to you now. All right. So non-functional, what type of solution do we have? This is a hypotonic solution. All right, so how many of you got that? Raise your hands. Wave your hands so I can see you. Very good. Most of you got it. Excellent. So that's test tube number 1. If you put this solution underneath the microscope, you would excuse me, you would see nothing. There would be no cells left. You would see absolutely nothing. All right. Let's skip the second one, because I think you probably have already figured out what's going to happen in the second test tube. We'll come back to it. But let's go to test tube number 3. And in test tube number 3, we had... I've already lost my place here. Ay, ay. What did we have in test tube number 3? Ay, 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 ay. Oh, lordy, lordy. Please forgive me. I'll get it. Don't worry. I'm there. I'm there. No, I'm not. I'm not there. I'm not there. It's probably in here someplace. Please be here. Please be here. Please be here. I don't know. All right, I lost it. Test tube number 3, we had a 9% solution. Okay, so I had 9% here. What do I have on the inside of the cell? The same as we've seen before, 0.9%. Okay. Let's do our analysis. We have 100%. We have 100%. Over here, on the inside of the cell, 0.9. On the outside, 9%. So this is 99.1%. And this is 91%. Is there a difference? Absolutely there is. That has a higher concentration, so water is going to move in this direction. It's going to move from inside the cell to outside. It's going to move in this direction. The cell is hypotonic. The solution is hypertonic. What about cell shape? Crenated. Remember, that's the term. We don't want to say shrink or shriveled because we don't sound very smart when we use those terms. And we want to impress people, so we're going to say crenated. What about function? Non-functional. Non-functional. And as a result of that, what we can say is that this is a hypertonic solution. Hypertonic solution. These cells will look like this. They'll shrink and they'll get all these little spiky things sticking out. Now imagine 100 million of these cells all kind of mixing up together, and they all have these little spiky things. So when they do that, these little spiky things will interact. They'll sort of interdigitate. And so it'll be much more difficult for these cells to slide past one another because of these it's like gears. They'll be like this, okay? Interlocking. As opposed to a normal shaped cell, which has these very smooth edges. So when two cells come into contact, very easy for them to slide past one another. So let's come back to our observations. And that's, of course, the page that I lost. But remember, I said in this cell, you would see, or in this test tube, you would see that the cells stuck together more. They were thicker. And the reason it looked thicker is because the interaction of the blood cells were such that they didn't have a chance to move past one another. They would be stuck to one another. And as a result, they have more viscosity to them. Okay. So I'm still looking for that. What an idiot. I don't know where it goes. Things just kind of disappear around here, and I have no idea where they go. All right. Be that as it may. These are non-functional cells, and certainly something that you don't want to happen in your body. Okay. Let's talk about the next or last experiment, which was the, here it is, okay, which was this, physiological saline. So physiological saline. And you probably have guessed now what that's going to look like. So that means that this is 0.9%. This is 0.9%. And I think now you can see that there's going to be no net movement of water. And the cell shape will be, this is a isotonic solution, or physiological saline, if you will. Cell shape will be normal, and we'll have functioning cells. We will be able to maintain homeostasis in these conditions. Okay? All right. So at this point, I want to say something that might help you. There it is. Oh, silly. It's stuck way in the back. Okay. So at this point, I want to say something about the terminology. Isotonic, hypertonic, hypotonic. Let's get those terms up here. Isotonic, hypotonic, hypertonic. These terminologies describe different kinds of solutions. And when we're talking about osmosis, we are trying to figure out where water is moving. We're trying to figure out which solution has the highest concentration of water. But notice that the terminology we are given to describe those conditions causes us or makes us focus on the solutes, not the solution. Isotonic means same number of molecules. Hypotonic means less number of molecules. Hypertonic means more number of molecules. It says nothing about the water, the concentration of water. So it is or becomes a little bit like a mental gymnastics to try to figure out which one of these conditions, when you're comparing them, has more water in them. Isotonic is not a problem. They're the same. But when you're trying to figure out if water is moving into a cell or out of a cell, the terminology you're given is not very helpful. You have to do a two-step process. Hypotonic means less solute. Less solutes means more water. So it's a kind of a two-step process before you get to the answer. And as long as you're aware of this, then you can sidestep it, realize that that is a stumbling block, and prevent yourself from falling over with the terminology. Okay? All right. So let's come back, if I haven't lost it, which I haven't. Okay? Let's come back to the end of the study guide, and you'll see a number of questions that I've listed here. In fact, there are a total of 24 questions. Okay, so starting off with actually 9, so we have less than 24 questions, but from 10 through 24, I have a number of questions. And I want you to, after seeing this video, I want you to take some time and answer these questions. I would suggest either having 2 copies, and 1 copy you can write on, and the second copy, when we get together in our Zoom meeting, we'll go over and through these questions together to make sure everybody understands what has happened. So this will be the subject. The answering of these questions will be the subject of our next Zoom meeting. And then I'll open it up to whoever has questions or follow-up questions or anything that you want to clarify in this presentation. So that concludes the presentation for Lab Study Guide number 2. And I will see you back here for Lab Study Guide number 3 when we begin to talk about tissues. So adios, amigos. And we're out.